It's like it has to be like that. I don't know.
That's weird.
Definitely weird. Okay, hi everybody. Miss Swantek here.
And Penelope.
We are going to be demonstrating part 4 of your blood lab, Predicting Inheritance. So, like we learned in class, there are 3 alleles that determine blood type. And within the human population, those 3 alleles, IA, IB, and little I, exist in different frequencies. Okay, so they're not all equally represented by the same amount in the gene pool. Okay, so our container here is representing the gene pool. And in the human gene pool, that means all the alleles for all the humans on this planet. Okay, for all the alleles, for all the humans on this planet, this is the frequency that we find these alleles. Okay, so the little I allele is most frequent, meaning it has the highest frequency. So 60% of alleles are little I alleles in the human population. 10% of alleles in the human population are IB alleles, and 30% are IA alleles. Okay, so the least frequent allele in the gene pool is IB, okay, and the most frequent is the little I allele. Okay, so our gene pool contains different alleles on each of these cards. Okay, and this gene pool reflects these frequencies that we find in the human population. Okay, so we have 6 times as many little I alleles as IB alleles. Okay, we have 3 times as many IB alleles. No, we have 3 times as many IA alleles as IB alleles. Okay, so what we are going to be doing is we are going to demonstrate the procedure of how you would do this if you were in class, and then we will run through the results. So we'll demonstrate the results as well. Okay, so we'll do 2 practice, okay, and then we will do, how many?
5.
5 collecting results, and those are going to be recorded in your lab, okay? So, make this smaller. Okay, so I am going to be parent 1.
And I'm going to be parent 2.
Okay, so I'm going to choose 2 alleles from the gene pool to represent my genotype.
And so am I.
Okay, so this is my genotype.
And this one's mine.
IB little I. Okay, so my phenotype is type B. Okay, so I'm type B, but I'm heterozygous B. Okay, but my phenotype is type B.
My genotype is IA, and my phenotype is homozygous A.
Okay, now, by the process of meiosis, when I make my gametes, because I sexually reproduce, when I make my gametes, randomly, these 2 alleles separate and end up in different gametes. Okay, so my gamete, my egg or sperm, gets this allele.
And my egg or sperm gets this allele.
So if this, say I'm the male parent, if this sperm fertilizes that egg, our offspring's genotype is, Penelope?
Our offspring's genotype is IA little I.
Okay, so this is our baby. What? Anubis? If this is our baby, okay, then our baby's blood type is?
A. Heterozygous A.
Heterozygous type A. Okay? All right, that was a demonstration. We're going to do one more demonstration. This is not for our results yet.
So don't record this.
Do not record this in your data table.
Well, we're recording it. Well,
don't confuse people. Mix that up, please. Okay, so now, again, I'm parent 1.
And I'm parent 2.
I'm going to choose my 2 alleles.
Oh Homozygous A.
So my genotype is IAIA, and my phenotype is type A. I'm heterozygous type A. Penelope?
No, you're homozygous type A.
I'm homozygous type A.
So my genotype is I, and I am homozygous type O.
Okay, now we're going to randomly separate our alleles, okay, into our gametes. And if these gametes come together in fertilization, our baby
has a genotype of IA, and our phenotype is heterozygous A.
Very good. Okay, that was our 2 demonstrations. Now we're going to do it 5 more times, and these are the results that you're going to record in your data table.
But we're not going to say anything.
We are not going to tell you the genotypes, and we're not going to tell you the phenotypes. Okay, here we go. So this is our first round. Get our baby. And this does he have a face? Oh my. And this is our baby.
That was
All right, that was our first one. Okay, remember, I'm parent 1 and Penelope is parent 2. I'm parent 1.
And I'm parent 2.
This is an easy one. This is our baby.
The only human thing.
Okay, this is our third one.
Yeah.
We should be recording this.
We are.
Oh, for goodness sakes. I'm parent 1.
And I'm parent 2.
Hold it up there a little longer.
I'll show them mine in the document camera.
Okay. Get our baby.
Let me this again.
This is our baby.
And it's a jackal.
Looks so good. Oh, for goodness sakes.
Really? Really?
I wonder why this keeps happening.
Yeah, me too.
Really? Are you really wondering why?
No.
I'm parent 1.
And I'm parent 2.
Get our baby. And this
Well, how'd you get a half lion, half eagle?
is our baby. Okay, this is our last one.
Last one.
Pick something good this time, Penelope.
There you go, Troy.
Okay, I'm parent 1.
And I'm parent 2.
Making our gametes.
Finally something other than...
Where's our baby?
Oh! Yeah. Yes, we are Nationals fans.
And that is our baby.
I had to do something funny. It was our last one.
Okay, so in your table, you would have recorded the genotype and phenotype for parent 1, the genotype and the phenotype for parent 2, and then the genotype and the phenotype of the offspring.
And we did not say them. You have to look at what we presented.
Good luck. Bye.
Bye.